Sylvine, it's up to you now.
Okay, so good afternoon. Welcome everybody to a new session of the colloquia organized by the Latin American Consortium for Nonlinear Dynamics and Complex Systems. This time we have talks by two people from the University of Buenos Aires. First, we will have Alejandra Ventura from IFIBINE, which is the Instituto de Fisiología, Biología Molecular y Neurociencias, a UBA and CONICET Institute in Buenos Aires, Argentina. Alejandra did her PhD at the University of Buenos Aires. Then she did a postdoc in Michigan and Ann Arbor. And then she came back to Buenos Aires, where she's now a professor at the physics department and a researcher at this institute I just mentioned. Today, Alejandra is going to talk about pulsatile inputs in cell signaling, frequency, selectivity. So thank you, Alejandra.
Thank you, Silvina, for the invitation. So let's start. This is a picture of my institute, which is in Buenos Aires, and these are the members of my group. Candela here is a PhD student in the group, and today I'm presenting mainly results obtained by her. And so to start this talk, I selected 3 keywords. The first one is mathematical models, the second one is frequency selectivity, and the third one is time windows. Let's go with the first one. Mathematical models is what we do in my group, mainly with focus in cell signaling and gene regulation. And we do it because we believe a correct model helps to understand the underlying mechanisms, and also the model can predict the system's behavior under various conditions. And I leave here a very interesting reference by Ray Goldstein about the how useful or valid, let's say, are theoretical results in biology. The second keyword is frequency selectivity. To introduce that, I selected this video. So actually we are going to see 2 videos. What we see there are yeast cells, which are eukaryotic cells, unicellular organisms with some signaling pathways that are common with mammal cells. And so these 2 groups, which are in the same initial condition, will receive an osmotic stress. The group in the left will receive a constant osmotic stress, while the group in the right will receive a periodic stress, with particularly in this case, a period of 8 minutes. So even though I won't explain the details of what's going on, of what means yellow here, we see that we end up with much more cells here than here, and the shape and the size of the cells is different here. Actually, they are not very healthy. They are about to die here. So this was taken by an article in Science from some years ago. They did this experiment, the one on the right, several times with different periods, and they quantified 2 properties, at least 2 properties, the generation time and the cell area, and the 2 of them have a clear maximum at 8 minutes. So this work was an inspiration for us. We started to think about selectivity and how could be that the signaling pathway could present this type of responses as a function of frequency or period. And so going a little bit theoretical, whenever we do a model, we first try to tackle the input-output curve. Let's say we apply an input that is constant in time, and we select one particular property of the output. for example, as in the case I showed, the area of the cell. Then we change the property of the input. The only one we can change here is the amplitude, the strength. We measure again and again and again. We get this input-output curve. Now, if the input has some temporal structure, like here, there are different options to control, like the duration of the pulse, the time between pulses, the period or frequency, the amplitude. In the talk today, I will be paying attention to output as a function of frequency. We have all these possibilities. The first one is what is called an all-pass filter, meaning that the system doesn't really care about the frequency of stimulation. This one is a low pass. If you stimulate at a higher frequency, the system responds less and less. The opposite, a high pass. But I will be paying attention to these cases, a band pass where we have a maximum, or the opposite could be band stop. And if that maximum is very narrow, we say we have a high selectivity. So there are different examples in cell signaling of these different cases. Here I selected an example of this case, the all pass. So in this case, they were studying calcium oscillations, and these oscillations were activating a transcription factor called NFAT, and somehow they could control the period of the oscillations in low, medium, high, and they measured the expression of NFAT, which is kind of constant. So they concluded that NFAT is an integrator of the calcium load. A different situation is this other case here, is a paper of mine from some years ago, where we were studying the nitrogen assimilation control network in E. coli. And we did experiments and modeling with higher and higher frequency until the case where there was almost no response here. So this is a low pass filter. And this other article, more recent, they control how ERK oscillations control gene expression, and by an optogenetic device, they have some control on the frequency of ERK oscillations, and they found at least 3 genes that express better at a particular frequency. So of course, I'll come to this again because that's the main topic of the talk, but let me introduce the last keyword, which is time windows. So let's say we have our cell, our test tube, our portion of a signaling pathway. We apply an input, we measure an output. So if the input is constant, then we'll get something like this, an input-output curve. If the input is pulsatile, we could get, for example, something like this. Our work in the past years have shown that both type of curves, input-output versus input's amplitude, for example, and output versus input's frequency, are really dependent on the time window where we do the measurement. So let me mention a very simple example. If we consider a ligand receptor reaction, with in many cases is the first step in a signaling cascade, and then we control the complex between this ligand and the receptor. Let's say we start with no complex, we add an amount of ligand, and then the complex will follow these kinetics. If we add more ligand, then we have this curve, and so I'm showing here time courses for different input concentrations. Now we take each pair of the ligand we added and this value here, and then we create our input-output curve. Now, let's say that for some reason we cannot wait until this time to do the measurement because the ligand is removed, because there is a downstream component here that has a very fast time scale and will read the complex at an earlier stage, and then we do the measurement here. Again, we take each pair and we find that the input-output curve is this one. And if we do an even earlier measurement, the input-output curve is this one, and earlier this one. What I'm showing here is an input-output curve that is evolving in time. And this was done analytically or numerically, but also some years ago, we validated this experimentally when I was working in Coleman Lerner's lab in the same institute where I work. We use yeast cells. The ligand was pheromone. The receptor was the pheromone receptor in the mating response of pheromone, and we did the measurement for different times, finding this behavior. So all that information is in this article here, and in a later article, we characterize the same behavior in different signaling scenarios, not just a ligand receptor one. And why this is important, this input-output curve tells us for which range of inputs the system will respond sensible. And so if we can do the measurement in a different time window, we can control the properties of this input-output curve. So basically what I'm saying is that if we stimulate constantly and we select where we do the measurement, then we can control the properties of the input-output curve. Now, if we do this type of stimulation, again, we can select where we do the measurement and what's going to happen. That's what is the talk about. So summarizing, mathematical models is what we do. We are paying attention to frequency selectivity and time windows. And basically our finding is that we could find a new behavior, let's say, that is this frequency preference that is only transient. And all the results were published last year in this article. I'll show you some parts of it, but so the main questions I will try to answer in this talk are these two. Is there an optimal activating signal for a particular target? That's the first one. And the second one is, do we need a certain level of complexity in order to get this selectivity, or are there minimal signaling components able to achieve those maximums? So what was known about it before we started working on this? Well, in these two articles and many others, we found that if you go too simple, like ligand binding, protein-protein interactions, covalent modification cycles, or cascades, the behavior is always a low-pass filter. But if we add a little bit of complexity, like a negative feedback loop, as we see here, or an incoherent feed-forward loop, then we can get a band-pass filter behavior. So our approach was, so let's consider very minimal signaling networks. In the example here, I'm showing a 3-node network. Node A is receiving an input, which is pulsatile. In node C, we are going to measure the output, where output means a mathematical operation over C. So we'll get C over time, and then we have to compute something else that is always given by the system we are studying. In some systems, it's important to compute the amplitude. In some others, the integral under the curve. In some others, the integral above a threshold. Okay, and in a very small system like this, with 3 nodes, we have 9 arrows. Each arrow could have 3 options, being positive, negative, or 0, according if it is an activation, inhibition, or nothing. And once we say, for example, it's an activation, we have to define the kinetics, what type of activation it is, and then the parameters. So even if it is very small, there is a huge computational space to explore. And we are going to define an outputs property and try to find out if there is a maximum as a function of frequency. And so the results are organized in 4 blocks. Probably I'll tell you about the first one and second one, and then just mention the last 2. So the first group is about ligand receptor again. The second one is about one-dimensional signaling nodes, then more complex signaling systems, and a little bit about experimental validation. So ligand receptor, I said before, no selectivity in this system. For some reason that I won't mention now, we revisited this example. So we have this reaction, receptor-ligand complex, then a simple model with an ordinary differential equation, the dimensionless model in which gamma is going to be the pulsatile signal, Y, the complex, the dimensionless complex is the output, and then there is a dimensionless time in terms of a reference time, which is the unbinding reaction. And then we apply this input. I want to mention that in most of our studies, we consider a dose conservation approach, meaning that if we are going to apply more pulses per unit time because we are increasing the frequency, we have to compensate either decreasing the amplitude or reducing the duration so that the dose given in the experiment is the same, so that we can identify an effect that is purely done by the frequency and not by the dose. And so I said the output is Y, and then the response has to be an operation over Y. I'm showing two operations here. We studied several of them. So the first one, called G, is the integral between a time S and S plus T. So the integral in one period of the input, normalized. This is what we call the sliding window. And H is the integral between 0 and T and S, and that's what we call the accumulating window. And so it can be shown that this system reaches, when receiving a constant step-like stimulation, will reach a steady state monotonically. We can obtain the time to reach steady state. It can also be shown that this system will reach a periodic solution when receiving a periodic input. We can estimate the time to reach that periodic solution. And well, this time depends on the frequency of the stimulation. Since we are going to be moving that frequency, we want to be something that is frequency independent. And so this time here is a good estimator to divide transient and periodic regimes. And so I'll show you now in one page the summary of several studies. Here, the first result is the normalized integral in one period of the input, what I called before sliding window. In orange, that was computed in the transient, and in green, in the periodic regime. And then we found that this quantity, the sliding window, exhibits a maximum in the transient, but not in the periodic regime. Doing the same stuff with the other variable, expanding window. And by playing with this quantity S, the upper limit of the integral, we can compute this quantity H in the transient or the periodic, and we find that there is a maximum in the transient, but not in the periodic regime. So this is showing us that the ligand receptor system exhibits frequency preference responses in the transient, but not in the periodic regime for these two cases and some others I'm not showing here. And this is what we call a transient frequency preference response. So these turn out to be a very robust behavior. It appears in the 4 combinations of the 2 quantities I mentioned, the 2 conservation protocols I mentioned too, where we compensate by reducing the amplitude when increasing the frequency or reducing the duration when increasing the frequency, and also happens with this for sinusoidal stimulation and without dose conservation. And so taking together this result with the previous result I mentioned about the ligand receptor system, we now have this pulsatile input. And typically it is a low pass filter, and different curves are representing different receptors. But if we go to an earlier time window, we can develop a maximum. And so this is a frequency selectivity that emerges depending on the time window and the response considered. And all our results are included in these articles here. And the meaning of this is that if we have a slow reader downstream to the complex, it will get something like this. A fast reader will get something like this. So the slow reader will respond to red frequencies, while the fast reader will respond to violet frequencies. And so this same device, a very simple binding reaction, could lead to very different information processing scenarios. So the second group of results is about a simple one-dimensional node in which we consider production and degradation. The production is a function of a pulsatile input, and this is already dimensionless. The output is Y, and the response, I'm showing the sliding window. And again, this reaches a steady state in a time 1 is 1 because this is already dimensionless and the degradation rate is 1. And then it can be shown that when receiving a periodic solution, this reaches, when receiving a periodic input, this reaches a periodic solution. We can estimate the time separating transient and periodic. And this tau here is a good estimator to divide the two cases. Now, in this system, I need to say which is the function f processing the pulsatile input. So numerically, we consider different types of monotonic increasing functions f that are relevant in signaling. A convex one, a linear one, a concave, and sigmoidal. And there are some examples here. And then we computed numerically the frequency response. Light colors are computed in the transient and dark colors in the periodic regimes. So we see there are no maximums here and here, convex and linear. However, there is a maximum in the transient stage in the concave case, and both in transient and periodic case in the sigmoidal one. And so because the system is so simple, we could do also some analytical calculations, provided f is derivable, positive in zero infinity, and then considering s, the time where this sliding window starts, s higher than 1 is the periodic phase, so we compute the derivative, and we want this to have a 0 so that it could have a maximum. And so asking for this to have a 0 defines the concavity of the curve. The sign of the derivative is given by the concavity of this function f, and then we could obtain the same results as before. The convex in the periodic phase is a low-pass filter, the linear is constant, as we see here, and the concave is a high pass, and the sigmoidal has a maximum. And then working with s between 0 and tau ss, we did the calculations in the transient phase, confirming that these two work as low-pass filters, and the other two, concave and sigmoidal, could develop a maximum depending on the function and depending on the parameters. And so what is f after all? What is interesting here is if we pay attention to the model, then f is the steady state level of y in response to a constant input. So if gamma is constant, then the steady state of y is exactly f. So this means that the form of the nonlinearity in the dose response curve under constant stimulation is predicting the existence of a selectivity when we stimulate with a pulsatile signal. I'm not sure if I'm doing well with time. Probably I have another
We
5
have like
minutes?
5 minutes, yeah.
Okay. So what we did was to find out if this selectivity could be transmitted to another component. So we took this level as before, we added a second level, Y2, and then there is a tau appears here, which is the relative time scale between Y1 and Y2. And again, F1 could be linear, concave, sigmoidal, whatever, the same F2, so there are many different options. And so to summarize these results, I'm using a color code. We have two one-dimensional nodes. I have to define F1, F2, tau, and the color code is saying I use blue for a low pass, I use orange for a transient selectivity, and I use green for a bandpass filter. So if we couple two blue nodes, the network, this small network, will be blue, low pass. If we have a low pass upstream to a bandpass, we remove the maximum, the network will be blue. But if we have a transient node here, a transient maximum here, coupled to a low pass, this can be transmitted provided that tau is small, meaning that this node will read the information of the transient in this one. And then we added some feedback to the model, the same model as before. We added this term here, trying to find out or to confirm what we knew from the literature, that selectivity could emerge as a network effect. So again, I take the same color code. I have to define F1, F2, tau, and delta, the strength of the feedback. And anything could happen here. The feedback could develop maximums, even in a completely blue network. A low-pass network with feedback could have selectivity. So after that, we studied very well-known models like the MAP kinase cascade, the gonadotropin-dependent ERK signaling, and studied if this could have transient maximums when stimulated with the pulsatile signals. I'm not showing those results now. And I just want to mention briefly that we are also working on the experimental validation of all these. We are working in a particular system that is called microRNA-mediated regulation. You know that from DNA, you go to messenger RNA, then to protein, but there's something here, a level of regulation at this stage that is done by microRNAs, which are very short RNAs that don't code for proteins, and it inhibits this step. And so we are doing some modeling on that. There is some binding between these two, the messenger and the micro, and then some recycling. And so the numerical results are in these two articles here. We found that the quantity of interest here is the fold repression, how much you can repress the production of protein. And that fold repression is better for some particular frequencies of the synthesis of this microRNA. And that's numerical, and we are doing some experiments on that. They are not ready to be shown completely, but we found that by controlling the microRNA pulsing frequency, there are some intermediate frequencies in which the inhibition, the fold repression is better. And I think that's it. We did also some analysis on published data. In this case, we were considering P53 tumor suppressor gene that typically oscillates and controls gene regulation. And in this very nice paper, they control the frequency of P53 in low, medium, high. And what you see here are expression of one particular gene in single cells. That's why it's so noisy. And so we analyze all this data in an undergrad thesis, finding that in some windows there is some selectivity. So the expression is better for the intermediate frequency, and if we change the window, we can lose that selectivity. So the summary, we studied very simple signaling components. Then we studied if this selectivity could be transmitted, could be modulated by targets. We studied some, I haven't shown those results, but we studied more complex signaling models. And mainly is this frequency preference response was overlooked in simple systems because of its transient nature, meaning we have a signaling component C, and if the signal activating C is short compared to its time, the time scale of C, we'll get a response like that. We evaluate this response for different frequencies, and we got a maximum. While if the signal is long enough to activate this type of responses, then probably depending on the complexity of what is C, we can lose that selectivity. And to end, I want to mention the funding, the lab members. The results were obtained by Candela Sitchik and a former member of the lab, Juliana Reves. The experiments are done in collaboration with this Italian group, Carla and Elsie, and all the mathematical stuff I presented was done in collaboration with Costanza and Rocio from the math department. Thank you.
Thank you, Alejandra. Very interesting. I don't know if there are questions. I don't know how I see if someone wants to ask a question. Okay, so let me ask a little bit something. Oh, there is someone asking?
I don't know.
Okay, so
what camera?
Who is it? I don't know. I don't see who. Is someone wanting to ask a question? No. Okay. So let me ask you something. So what do you mean by selectivity exactly? Because you have these maxima there as a function of frequency, and that would be like the selectivity, I guess. What would that mean?
Well, it could mean different things, and depending on the pathway, on the case we are considering, but mainly it means that the system performs better for a range of frequencies that are not too low, not too high.
So it produces something, a better response, or more, I don't know, concentration of something that needs to be reduced, exactly. Something like that would be. And then what are the time scales that determine that selectivity? Like, for example, in the case of the binding and binding examples that you gave, you know, what about, you have several time scales there, the binding, the unbinding, and what is it that determines the selectivity?
In the case of the simple binding, the time scale that is important is the unbinding rate, right? So you could think that's the only time scale. Well, we have also the time scale of the input, which is the duration of the pulse and the time between pulses. In the examples I showed at the very beginning, when ERK oscillations were controlling gene expression, and there were 3 genes having a maximum, if you pay attention, the horizontal axis is not frequency, it's time between pulses. In several cases, what is important is the time between pulses. I'll explain that in a while. And of course, we introduce another time, which is S. Where are we going to compute for example, the sliding window? So there are different time scales, and it's the interplay between them. And so related to what I said about the time between pulses, I haven't mentioned that, but to start analyzing all this, we classified the signaling systems in 3 groups. according to how they respond when you give a constant stimulation. So the first group is that one responding monotonically, and I presented in this talk exclusively that group. The second group is the adaptive one, that one that even the stimulation is constant, they respond and then they turn off. And the third group are the oscillators. So I haven't studied oscillators. The second group, we studied that separately and is in a different paper. And in that group is essential the time between pulses, because it's the time you let the system recover. And so in that case, is the interplay between the time between pulses and the time, not the time it takes to reach the maximum, but it's the time to recover. If you select a good time between pulses, then you can have this maximum. I'm not sure if I responded. Yeah.
Yes. So I think that we should move on. I don't know if there is anybody else who wants to ask something. If not, we thank Alejandra again. Thank you very much. And
Thank you.
we are going to move to the next talk. So Facundo, if you want to share your screen, please, while I introduce you. Facundo Fainstein, he's a PhD student in physics at the University of Buenos Aires. He's doing his PhD under the advice of Gabriel Mindling on birdsong. And he's about to finish. He's already looking for postdocs, so that's a hint here. And he's going to talk about fine-tuning between neural and body dynamics. So yeah, the floor is yours, Facundo.
Okay, so hello. Thank you very much, Silvina. You can hear me well, right? And you see the full screen and everything.
Yes, I see everything.
Okay. So thank you, Silvina, for inviting me. I'm very happy about it. So yeah, so the title of my talk is Fine-Tuning Between Neural and Body Dynamics. And I'll start by very briefly introducing a little bit more about myself. So I'm originally from Bariloche, which is this town that probably most of us know in the south of Argentina, in Patagonia, a beautiful place that every time that I have the chance of saying anything, I tell people that have been there or have not been there to go any time of the year. Is beautiful. And then I went to study physics at the Universidad of Buenos Aires, and I am now finishing a PhD in physics, working under the advice of Gabriel Mindling. So very early in my undergrads, I got interested in biologically motivated problems and dynamics. So as soon as I could, I started going around Gabo, and everything that I will be sharing to you today is work that I have been doing with him. So starting from big questions, being from Bariloche, I like outdoors a lot. I like hiking and going outside. And it is very easy to go out and get basically amazed by nature in general, and I would say by animal life complexity in particular. And I think that it is amazing how animals can accomplish tasks, from the most simple one as to perform a single breath to the ability to do complex behaviors such as singing. And all this happens because of, or emerges due to the interaction of a very complex device, which is our brains, and also the interaction of this brain with a physical entity, which is the body. So there's a very interesting question that is, well, how come that by the interaction of these neural dynamics and the physics of the body that we gives rise to animal physiology and behavior? And we are going to go around this question in two stories, which could be separated as looking at animal physiology and behavior under the sunlight, and then under the more darker, misty, and less solid grounded periods of night sleep. So, being a physicist, we're interested, of course, in the fundamental laws and the fundamental building blocks. So, of course, we would like to go first of all to the brain, which is this beautiful device that we have. So we would like to start from the microscale. So this is what a teardrop of brain tissue in the brain of a mouse looks like. And this is what a single cell looks like. So one could say, well, I would like to start with a blank notebook and write a simple dynamical model for a neuron. I will have some parameters, and then I will have to, I will have thousands of cells, so I will have at least thousands of parameters, and then I will have to take into account that this is a biological neural network. There's some connectivities, so I may write a matrix that will tell me who's connected to whom. So I will have then, I will have to add order n squared parameters. And one sees that, of course, there are very, very interesting things that one can do and people have been doing with this approach. But there are also very interesting challenges that arise and very interesting questions that I think that are very informative too. So one of the questions is, well, how am I going to wisely choose millions of parameters? And maybe more interestingly, what is going to be the biologically relevant quantity that I will try to compute and I will try to measure? So one can get a very strong headache with this, and it is not easy to make sense of this. And I think that at this point it is good to remember a very famous quote in biology that is that if you want to make sense of biology, you should look in the right light, and that is the light of evolution. In particular, if we are thinking about what are going to be the parameters of a biological problem, these parameters will live within ranges, and these ranges are not anything, but these ranges have been shaped through millions of years. And this is true for the parameters that define the physical properties of the body, that could be the shape of a beak of a bird. And I think it is reasonable to expect that these parameters will also this will also be true for the parameters that define the dynamics of the neural circuits of our nervous system. So this idea raises another question, which is also, I think, very interesting, which is, well, to what degree these neural and these body parameters are attuned to facilitate physiological functions. So we study this question in the context of probably the most fundamental physiological function, which is breathing. And we study this using birds as animal models due to many reasons. Reason number 0 is, of course, that Gabo, as most of you know, works on the physics of birds for a long time. But I would say that there are other reasons, and one of the reasons is, maybe for your surprise, that people have been interested in the respiratory system of birds due to their anatomical complexity, and because it is believed to be a very efficient system, and because it is believed to be a system that has allowed birds to have the success that they have, that it is basically that they can live in every place of the world, they can fly, swim, migrate thousands of kilometers in a couple of days, and do complex vocalizations such as singing. So this is what the respiratory system of birds looks like. They have lungs, like we do, but in contrast to our system, these lungs are rigid, and the ventilation is achieved by the active expansion and compression of a set of air sacs. So basically, during inspiration, the inspiratory muscles expand the thoracic cavity, so all air sacs are expanded simultaneously, and air flows from the outside to the interior. During expiration, expiratory muscles compress the thoracic cavity, so all air sacs are compressed simultaneously, and the air flows out. Actually, the airflow through the system is non-trivial, and they have a unidirectional flow of heavily oxygenated air through the lung in each phase, which is something that is quite interesting. And the other thing that is very interesting is the fact that when we look at what is the respiratory rate of a canary, that is the species that we study in our experiment, a canary would breathe when it is calm at 2 Hertz, which is this distribution in gray. And this rate, of course, is determined by the nervous system, which is sending periodically these motor instructions to inspiratory muscles and expiratory muscles. But this is not the only frequency at which this neural network can work. Actually, when they sing, the rhythm of the song is determined by the respiratory rate, and these frequencies can go up to 20-25 Hertz. So, given this, one of the first things that we learn in our undergrad studies is that when we have an electrical circuit, which is a resistor, a capacitor, and an inductor, and we force this system, there will be a frequency at which the current that flows through is maximized. So the question that we asked is whether the nervous system is forcing this biomechanical device at a frequency at which the airflow that ventilates the lung is maximized. So to answer this question, we developed a simple model for the respiratory system. Basically, what we have is that the movement of the thoracic wall is going to be the movement of a piston that when it moves, it changes the volume of the cavities that represent the air sacs while leaving the volume of the cavity that represents the lung unchanged. We write Newton's equation for the displacement of the piston, and we write a dynamical equation for the pressure of air inside the air sacs. The force that the muscles do is introduced in our model as an external forcing, and then we have 4 parameters. So given this model, we developed the techniques and we did the experiments to actually measure these variables. So we measure the pressure of air inside the air sacs, we measure the displacement of the thorax, and we measure the muscle forcing. We measure the muscle forcing by doing an electromyographic measurement, which is basically to measure the electrical activity in the muscles. What you do is you take 2 wires, you take the coating on the tip, and you place it in contact with the muscle. Basically, when the motor neurons are going to be firing, you pick up this voltage that will tell you that the muscle fibers are going to be contracting and there will be a force exerted on the system. Okay, so what does our data look like? So this is the dynamics during quiet breathing. This is the displacement of the thoracic wall as a function of time, and this is the pressure of air inside an air sac as a function of time. So this is a 2 Hz oscillation that we were mentioning before. And we see something that is, I think, very cute, which is that basically the system is a monotonous compression and expansion of the air sacs. But the dynamics of the pressure is non-trivial. Non-trivial in the sense that if one think of an air sac as being a balloon, a closed balloon, we would expect that when you monotonously compress it, the pressure would increase. And when you expand it, the pressure would decrease. But of course, these air sacs have an opening or openings. So what we see is that basically when you're compressing, the pressure increases, but then when you're starting to lose molecules, you continue to compress, but the pressure starts to go down. So, and this is actually what makes the dynamics of the problem non-trivial and actually makes the problem interesting, this non-trivial relationship between the pressure and the volume in the sacs. So we can fit these 4 parameters in our model to account for the dynamics of quiet breathing very well. So the question is, well, what happens during song? So during song, things get more interesting. So this is the spectrogram of the song of a canary. This is the frequency content of the sound as a function of time. I will try to reproduce this. I'm not sure if you will listen, but maybe you tell me, but I will put play.
Yes.
Yeah, okay.
Yeah, we could hear the sound. I mean,
Okay.
there is a lot of silence in between, I think. Is it? I mean, it started okay, and then no.
Yeah,
Okay.
for me, I don't know. We tried before, but anyway, I think you probably have heard a canary before. But anyway, so the thing is that this is the song, and to give some context here, when you start to think about this, it is very complex to achieve this level of complex vocalization. So I can barely speak, I do not sing, and it is not easy to get this. And people have been interested in birdsong for a long time, mainly for one reason, which is the fact that, which is actually a gift from biology, which is the fact that from the 10,000 species of birds, there are 40% of the species which have vocal learning, which is something that is not common in the animal kingdom. Canaries are among these. And then you have many species which do not learn. So it is a very, very interesting field to study what happens, or what are the fundamental building blocks of, or the fundamental properties of a biological neural network to be able to learn, and what happens in the network when it is learning, which is of course not the same as what happens during backpropagation, right? So how are these sounds produced? So the brain will be interacting very fast and very precisely with the respiratory system, and also with a very specialized organ, which is the vocal organ of birds, which is the syrinx. And basically, they have a couple of labia in the juncture between the bronchus and the trachea. So what they do is they send very violent instructions to respiratory muscles to violently compress the air sacs to generate a pulse of air that will flow through the bronchus. They will pull the labia together, and the airflow will go, and the labia will start to self-oscillate, modulating the flow and then producing the sound. So that is the basic idea. And so what do we see in our data of the respiratory dynamics during song? So it looks like this. This is again the spectrogram. This is the EMG, which is the electrical activity in the expiratory muscles, and this is the pressure of air inside the air sac. So this square wave oscillation of 2 hertz of the pressure now is transformed to these dynamics. And what you see is that each one of these sounds is produced by a very particular pressure pattern that they repeat with a marvelous precision, and that they have a very, very wide range of morphological pressure patterns due to the complexity of this song. I like to think of these two measures as being two thermodynamic quantities. The pressure is obvious why, but the EMG is also kind of, because we are picking up the activity of many motor neurons, and these motor neurons are kind of integrating, or the result of the activity of thousands of cells upstream. So here the interpretation is, well, when you see a electrical activity in the muscle, there will be a compression of the air sac, and the pressure will increase, and that is the basic idea. So we have this data, and we have our model that could account for the dynamics during quiet breathing. So the question is whether we can account for the dynamics during song, which is much richer. So I mentioned that we have 4 parameters. 2 parameters are expected to change during song. One is the amplitude of the forcing, and the other parameter is a parameter that tells how is the flow resistance in inspiration and expiration. And basically they expand the syringeal lumen during inspiration, and then they pull the labia together during expiration, so the airflow resistance is increased during expiration. So when we fit again those parameters and we use the EMG, the measured EMG as a proxy of the forcing, we can reproduce very well this wide range of morphologies in the pressure patterns. So here we arrive to a point in which we can go back to our question, because we have a model that is biophysically sensible and sensitive is always something that I get confused in English, but we have a biologically motivated model which can account for the dynamics during song and during quiet breathing, and our parameters have been informed by the data. So we can see what is the frequency response of the system by basically we take our fitted dynamical system, we change the forcing to be a harmonic forcing, and we see what happens to the pressure amplitude as we change the forcing frequency. And what we found is that the pressure of air inside an air sac displays a resonance, and that the resonant value is exactly 2 hertz, which is the frequency at which these birds breathe. What this tells you is that basically given amplitude of a motor instruction will be efficiently translated to a pressure pulse, and that is good because this pressure pulse is what drives the airflow that ventilates the lung. During song production, this system is being forced at a higher frequency, at a higher amplitude, and because the system is nonlinear, this is not just a trivial scaling of the resonance curve, but the frequency response goes towards higher frequency values, and the bandwidth increases in such a way that basically all syllabic rates, all the respiratory frequencies during song can get efficiently amplified. So what we found is that birds breathe and sing at resonances of the biomechanics, which reveals a shared optimization strategy across these behavioral states. And I think that something that is very interesting, which is that it shows a very intimate relationship between the parameters that define the dynamics of the neural motor circuits and the parameters that define the physical properties of the body. So going back to this question of how do we choose millions of parameters of a biological neural network, and what are going to be the biological relevant quantities will we predict and measure? Of course, I don't have an answer, but I think that given this work, I would say that there are key clues in understanding of what is the nervous system output and how this output is then translated to the physiological variables that determine behavior. In particular, we saw that the neural parameters are going to be related to the physical properties of the body. And then there's another thing that maybe it is too provocative, but it could be summarized as in neuroscience, people, we're interested in understanding the brain and what is the neural code. And one could say that the body is at every instant uncracking the neural code to translate these instructions to actual physiological variables or movement or behavior. So I think that there are a lot of things to learn about the neural code by looking at how the body is uncracking it. So I spoke for 19 minutes. It's a lot. I hope you're not too bored. I will finish soon. But before finishing, I want to share you about this thing about looking at animal physiology and behavior during the night. And when I realized how much time we sleep, I got a little bit depressed, because we spend almost a third of our life sleeping, which is a lot. But of course, sleeping is important, and of course, and I think it's very interesting to see what happens when we sleep, because our neural networks do not stop. In particular, we were recording this dynamics of respiratory dynamics during the day, and with Gabo, we decided to continue recording during the night to see what happens, and we would find the most boring thing, which is just this almost 2 Hz oscillation during most of the night. This is the pressure, and this is the EMG of the expiratory muscles. But we discovered that sometimes in the middle of the night, this activity would be interrupted by very violent activity in the expiratory muscles, and with this very slow 2 Hz pressure fluctuations would be interrupted by these very rapid oscillations, and with oscillations which had a morphology which was very, very similar to what we observed of the pressure patterns during song production. Actually, they look very similar, but the amplitude is very small. So they are not generating violent pulses of air, so sound is not produced. So we discovered that during the night, birds have song-like replay activity. This is something that was known, but we discovered that this goes down to the respiratory muscles. And Gabo, with Franz, one of his collaborators, had discovered before that this activity in the brain also goes down to the muscles in the syrinx. So we don't know why dreaming is important, but for the moment, what we can do is that because we know the pressure, air sac pressure code, we can associate the pressure patterns during the night sleep, during dreaming, with the pressure patterns during daytime singing to basically generate sound of this dreamed song, which otherwise would never be listened. And I think it will not be listened, but I will do play. You tell me, Silvina. Otherwise, I will finish by saying one thing. I'll say I put play. Silence, right?
Yes, nothing.
Okay. Anyway, last week we had Jose San Martin Day, and I heard that during the crossing of the Andes, San Martin, who was a very, very good musician, he took to the trip music by Beethoven, and he would had the Spotify of the epoch, which was just reading at the music and imagining what it sounds. So I will finish by just showing it in the screen. And thank you very much.
Oh, thank you, Facundo. Very interesting talk. I don't know if there are any questions. So apparently there are none. Let me ask you something about these resonance curves that you showed. Did you obtain them for a single? Is there someone who wants to ask a question? No. Did you obtain them for a single, like an individual bird, or is it from a collection? And I was wondering how much because you said it was like tuned to the body, how much it varies depending on the individual bird.
Yeah, so that's a great question. So we did a couple of things. In the case of this figure, it is showing the result for one bird. So with one single set of parameters, we can reproduce all the songs that we have, which are many. And we did the experiments with many individuals, actually 5 of them, and for all of them, our model works very well, and the parameters that we obtain from the data are very similar, and they always have this widening of the resonance curves during a song. Of course, it is not exactly the same for each individual, and there's this interesting question that you asked, whether these differences between individuals that we find in the resonance curves, which I'm not showing, but I tell you that there are something that is true of the biology or differences by the individuals themselves. And I think it's a very interesting question. I think it is not so easy to conclude individual differences from this. I think it should be possible, but it would require some solid analysis. Yeah, it is not easy because yeah, I don't know. I'm sure that the resonance exists. That is actually shown in yeah. That is true, and the widening is true. But the individual difference is something that is more difficult to conclude.
And then the other question I had was, I mean, for you to hear the song, the bird has to have its beak open. And you said that when they are asleep, the little picture that you show, it seemed like the beak was closed. And you said the pressure or the amplitude, I don't remember, it was so low. I guess it was the pressure. And that's why you cannot hear. And I am wondering whether the beak is opened by a separate set of muscles, or if it's open just because of the pressure of the flow that needs to go out. And I was wondering, maybe you don't hear because the beak is closed.
Yeah, that is very nice. So I don't know the muscles of the beak. What I do know is that in order for us to know that the birds were actually sleeping when we were recording this, we had a camera pointing at it to see that the eyes were closed, and we saw that actually when these song dreams happened, sometimes they did some sort of complete display of the body. So you would see that the bird would have like the head kind of pointing down when it was sleeping, and then when it was this activity, the head would kind of pull up with the eyes closed, and the beak would move a little bit. So and actually, yes, we measure also the respiratory activity and the activity in the syringeal muscles. So we know that there's a kind of a complete peripheral activation during these replay activities. But this, yes, also because of previous work of Gabo, we know that the oscillations in the syrinx to actually produce sound are born when the pressure is enough to make the labia oscillate. And you can maybe see here that these pressure fluctuations during quiet breathing, I don't know if you see my pointer, but then when
Yes.
this replay activity happens, the amplitude is very small, very similar. So that is not enough to produce the oscillations, to produce enough flow. So that is the reason why. And during song, the amplitude is increased like 15-fold what is during normal quiet breathing. So yeah, so this is very small.
Thank you. Hilda.
Yeah, I mean, part of what I want to know, you've answered, but I wanted to know when you measure the activity of the birds when they're sleeping, how do you measure the brain activity? Because you measure the fact that it's awake, close the eyes, etc., etc. But I want to know how you measure the brain activity of the birds.
Okay, thank you, Hilda. So I always did experiments by measuring activity in the periphery. So pressure of air inside the air sacs, activity in the muscles of the syrinx and of the respiratory system. But of course, activity in the brain can be measured, and that is something that an Amador does in the group. So during these experiments, we did not measure activity in the brain, but she does by recording in different nuclei associated with the song production part of the brain. So yeah, so it can be done. There's people that measure like global activity that tells in that can be used to separate what part of the sleeping periods you are, if you are in the REM or non-REM, these type of things. But what Anna does is, I think she's developing the techniques to do this also, but also to measure the activity in HVC and by measuring like many channels and with some electrodes
I think
placed in
they
the
do surgery and insert electrodes. Is that right? I mean, they do
Yeah.
surgery on the birds and insert electrodes. That's the way they measure, with electrodes that are inserted in the brain.
As they do it also with mouses, with rats. I mean, yeah.
Yes.
Yeah.
Yes, and it is, and you, sorry, yeah.
Okay. Thank you.
Okay. I
Yeah,
don't
you
know
can
if there are
imagine that
any
this.
Okay.
I don't know if there are any other questions. And oh, you stopped your sharing actually. Okay, I think there are no more questions. Let me run an ad before closing. I thank Facundo again for the very nice talk. And I just wanted to tell you, to those of you who are interested in biological physics, that we started a network in the physics of life of South America. And so far we've organized some talks, and we are going to have a talk next week on Friday by Pablo Marquet from Chile, very interesting on evolution and ecology, and at 11 a.m. our time, Sao Paulo and Buenos Aires. And so if you are interested, just send, and you haven't heard from the network, just send me an email and I tell you more about it. And I don't know, Albert, if you want to say something else, or if we close here thanking again via the two speakers and announcing the next session that is going to be about a month from now.
Silvina, thanks so much. Send the advertisement of your group, then we can put in our Instagram, okay? It's there. Thanks so much for all.
Okay, thank you. Okay, great.
Thank you.
Follow the Instagram thing.
Okay, bye-bye.
Bye-bye.
Thank you. Bye-bye.
Funciona.